To continue this conversation, and this really does fit the same theme, a few years ago, we launched what we called the Treatment Accelerator Program. And the idea was very simple, that if you do have a drug, a hypothesis for something that works, it really helps to have an infrastructure so that you can test it in multiple iterative systems. And essentially, I think I heard this metaphor from somebody else. If you stack pieces of Swiss cheese next to each other, you want enough so that if something isn't working, it's going to get stopped at a stage. And so we wanted to really be able to test drugs multiple times, and we wanted to do it in a way that was informed by our community. We wanted to kind of be in the kitchen cooking and trying to figure out ways to test these new drugs and set up a system to do that. And so we found Newsight, which was a contract research organization that was willing to set up a CAN model in patient-derived cells. And when we first started the relationship, it was a very exciting thing, but it's really grown into something else because Newsight has gone above and beyond every single time to make sure that their model systems are as accurate and comprehensive as they can possibly be. And our next speaker, Dr. Wayne Poon, is somebody who is always exciting to meet with. We always come out of our meetings with Newsight very excited, and he's the kind of person who will send you a paper at midnight because he saw it and thought that it might be relevant to something relating to KIF1A and CAND. So we're very excited to have Newsight represented here and very excited to hear from Dr. Poon about the Newsight platform.
Okay, well, thank you, Dylan, for the great introduction, and then also for the great talks by both Wendy and Simran. So, so we are a small biotech company located in Mountain View, California. Our founder and CEO, Tao Wang, he has a commitment and an interest in studying epilepsy and seizures and looking for a drug that treats epilepsy, because he has a personal stake in this. Okay, and so Tao is also a aficionado of classical music, and so at Newsight, what we think about the brain or the CNS system is it's an orchestra, and this orchestra is composed of the many different players of the brain, which includes neurons, but in addition to neurons, there's also other cell types. Glia support cells such as astrocytes, microglia, oligodendrocytes, and endothelial cells. And the concept is that the brain needs all these players or all these cell types to come together to play a piece of music and function properly. And in the case of CNS disease, neurodegenerative disease, and such as CAN, you have an out-of-tune orchestra. And the challenge for pharma is to develop a way to identify the out-of-tune player. In a case where, for the most part, we're tone deaf. We really don't know exactly what is going on. We know that we have these clinical symptoms like seizures, spastic paraplegia, but we really don't know how to define the cell type or develop a cellular model to then screen for compounds. And so KIF1A.org reached out to Newsight, given our expertise in building some of these models. And then I hope to share some of the progress that we've made over the past year and a half. And so there's two types of, I guess, phenotypes or defects that we can observe when we take the cells of the brain and we look at different mutations like KIF1A mutations. Potentially, they're called cell autonomous, or phenotypes that you see in a single cell by itself, or what we have a second phenotype, which is non-cell autonomous phenotypes, which is a phenotype that you only observe in the context of the other cell types. And so I think what we found is that these non-cell autonomous phenotypes are very important, but there are also cell autonomous phenotypes that we can use for drug screening. Okay, so this is just a timeline of where Newsight has started. We launched in 2015 as a biotech company, and this is placed within the context of different discoveries related to stem cell biology research. And as Simran alluded to, utilizing iPSC or induced pluripotent stem cell technology, that is really the foundational technology that NuSight employs. This technology started back in 2006 when Shinya Yamanaka identified factors that can reprogram your somatic cells, basically any cell that you have, your skin cell or your blood cells, and reprogram it back to a stem cell state. The hypothesis was that at fertilization, you have an embryo in which can emerge all the different cell types of the body. And then the idea is that it has the instructions for all the different cell types. And so then the terminally differentiated or somatic cell should have the ability to go back to a stem cell state. So he identified these factors and he won the Nobel Prize for this. And so since that time, there have been a number of advancements in the area of stem cell biology that identified the different factors needed to convert the stem cells to all the different brain cell types of that we see in the brain. And this will now facilitate disease modeling and allow us to study diseases in a dish. And so that's the strength of NuSight, is being able to develop disease models in the dish to then develop employ drug screening platforms to identify compounds that can rescue these phenotypes. Okay, so since 2015, NuSight has worked very hard on identifying ways to generate these cell types in volume and in scale with high consistency. And this is important for drug screening, that you have consistent cell types that you can work with, and so that with each batch, you're going to get reproducible results. And this is something that we have been able to achieve. Okay, so with this in mind, over the close to a decade, NuSight has been able to combine the different cells of the brain, the neurons, and then the glia, and then combine them to then identify phenotypes. As Simran alluded to, the electrophysiological phenotypes that can be captured with these devices that have electrodes that kind of mimic the EEG recordings that you see when you put electrodes on the brain. And then it is this platform that NuSight over the past decade has made quite a bit of advancement in developing seizure-based models. Okay, and these models, as I will go into shortly, are both in 2D and in 3D, and there's a rationale, as Simran alluded to, as why we are looking to develop 3D models moving forward. And so briefly, this is just the data that we've generated in the past year in our sponsored research collaboration with KIF1A.org. The foundation, again, is on our technology, in which we've identified the key factors in neurons that then make stem cell neurons. And then when we provide them to the stem cells, we can convert them very rapidly to neurons in a very quick amount of time. And then we can then combine these neurons to mimic the environment of the brain, and then recapitulate or phenocopy the phenotypes that you see in the brain. And so our standard platform is to take both the neurons and astrocytes, as you will see shortly, and combine them and then study the electrophysiological properties of the brain that can usually capture phenotypes, including seizures. Okay. And so in addition to the MEA and the electrophysiological phenotypes that I will describe, we worked with KIF1A.org to develop additional assays. And this really was a synthesis of a lot of the work that was that had been undertaken by the KIF1A Research Network. They've identified a lot of aspects of KIF1A biology that then allow us to translate the biology to screens. Okay, and then there are a couple other assays that I won't touch on as they are still in development. But one assay is the RNA-seq analysis that we will be embarking on, and that involves isolating the messenger RNA, which are the transcripts that are encoded by the DNA of all the gene expression transcripts in a cell, and this can identify perturbed or altered pathways due to the KIF1A mutations that then can inform on potential drug targets. And so we think that this is a exciting path or novel pathway to identify compounds, in addition to the repurposing effort of Australia Group. Okay, so this is just a representative picture of some of the neurons that we can generate from CAN patients. And so in collaboration with KIF1A.org, they provided us 2 patient iPSC lines from the P305L and the E253K mutations. And these were strategically chosen given specific phenotypes that manifest clinically. Okay, so the first assay that we tried to develop was an MEA-based assay, in which we took the neurons from the wild type or the patients with the KIF1A mutations, and we combined them in a dish. And then we just watched them mature over time and measured the electrophysiological properties. And using this MEA or microelectroarray system, you can capture up to 24 different parameters. And then using a bioinformatics tool developed by Massaro et al. in 2021, and he is a scientist at Axion, which is the MEA company, or the company where we get the MEA devices from. We can take all these different parameters and kind of develop a linear relationship or a two-dimensional representation of the large data set of the 24 parameters. And using this this score, which he called the neuroactivity score, we could capture a developmental phenotype, i.e. that is the KIF1A mutants, E253K and the P305L, exhibit a neurodevelopmental phenotype or that is different from wild type. That means they mature later, or they don't mature as quickly as wild type neurons. And so this was very exciting to us, given the clinical observations that CAN patients often have a developmental delay. And so the idea that we can take the patient neurons and identify a similar phenotype was something that we thought could serve as a potential drug screen. Now, one of the parameters, one of the 24 parameters that showed quite a bit of significance was this called network burst frequency, in which all the neurons that are cultured in a dish fire simultaneously, and over time, what we observe is that we can recapitulate again this neurodevelopmental delay, or we see this decrease in network burst frequency across early development from day 0 up to day 35. In fact, we see this delay up to about 100 days. Okay. But something curious happens around 100 days in that we see a shift in activity, and that the neurons that possess either a E252K or a P305L mutation now show an increase of network burst frequency. And this is essentially a phenotype, a seizurogenic phenotype in a dish. And so typically when you see increased network burst frequency, that is associated with a seizurogenic phenotype. And that is something that we can observe in a dish. So this interesting phenomenon in which we see at first this neurodevelopmental delay, then followed by a seizurogenic phenotype in a dish, seems to coincide with some of the clinical observations in which, you don't often get, or you get a delay in seizures in many of the CAN patients. And so it was very exciting for us that we saw this, that we were able to recapitulate this in a dish. So the next thing that we looked at was development of a three-dimensional model of the brain. Now Simon talked about a method that they're using in Australia where they're taking the stem cells and then taking them all to brain organoids over many, many months. And so that sometimes is not practical for drug screening. Okay. And so here what we're showing is a three-dimensional what we call organoid or assembloid, in which we've combined mature neurons, the neurons and the astrocytes together in 3D, and that we can recapitulate this seizurogenic phenotype in less than 30 days. Okay, so if you recall from the earlier slide, we see more of a neurodevelopmental delay prior to 35 days, and then it takes about 100 days before you see a seizurogenic phenotype. Now, when we take these cells and put them in a 3D context, we can accelerate the seizurogenic phenotype. And so this is a very exciting development, because in a parallel effort for some of our other neurodegenerative programs, we've been working with a biotech company called Maxwell Biosystems in Switzerland to develop a microfluidics platform to screen organoids, which involves their high-density MEA platform. And so we think that we can apply this technology or this microfluidics device to then utilize the seizurogenic MEA phenotype to screen for compounds that can potentially reverse this seizurogenic effect. Okay, so these are the phenotypes that we observe when we take the neurons and we combine them with the other cell types in the brain, in this case astrocytes, and we can observe these electrophysiological property differences. And so now I want to shift to some phenotypes or cell autonomous phenotypes that we uncovered in just looking at the neurons by themselves. And so we use this camera, which can take pictures regularly spaced and follow the development of the neurons in the dish. And what we observe is that we see again a deficit in the neurons from the CAN patients compared to wild type. And there's a number of metrics here that we see deficits in, including neurite branch points per soma. And so the number of neurites that emerge from the cell body is impaired in CAN neurons compared to wild type. And so this is something, or this is a phenotype that we can observe in less than 7 days. And so this is again parallels some of the clinical manifestations of potentially a neurodevelopmental delay, in that we think that we can utilize this imaging or modality to then screen for compounds, as we can achieve looking at a phenotype in less than 7 days. Okay, now this live cell imaging phenotype is corroborated with secondary validation, in which we then do some immunohistochemistry and identify deficits associated with the neurites. What we're working on is algorithms to also process and look at all the different neurite branches that emerge from a brain cell or neuron, and these are called dendrites, and each of these little morphologies indicate the types of synapses and the type of brain connections that a neuron can make. And we think that this might be indicative of the neurodevelopmental delay in CAN patients. And then this was something that this is an experiment or an assay development assay that we developed in collaboration with Inovio, which is another biotech company in Canada. And so from the KIF1A research network, we know that KIF1A is a motor protein that when it's mutated, it affects axonal biology. And so with this microfluidic device from Inovio, we were able to develop an assay to look at how axons grow in response to neurotrophic factors or growth factors. And we've identified a specific impairment in the brain-derived neurotrophic factor, or BDNF. This was specific to BDNF, as other neurotrophic factors, we did not see this impairment. And so again, with these devices, we can assess this phenotype in 7 days. And so this again is another promising assay that we have to then screen for compounds. And so at NewSight, with our commitment to identify compounds for epilepsies and seizures, this year we are planning to carry out a small library screen of 1,280 compounds, and we're utilizing a library called the LOPAC library. The reason why we're going to use the LOPAC library is for different reasons. It was utilized in a screen for Alzheimer's disease and had identified a number of compounds that affected microtubule dynamics. And so we know that KIF1A is a motor protein implicated in microtubule dynamics. And so we think that this LOPAC library might yield compounds or similar compounds that would reverse some of the phenotypes that we've uncovered. In addition to this library, with partnerships from KIF1A.org, we have a number of other compounds listed here, and they also will target different aspects of axonal biology. And so we think that these are potential small molecules that may rescue the phenotypes that we've uncovered with the different assays. So one last comment is that Simran highlighted research funding dollars. We are a small biotech company. The support for all this work was provided by KIF1A.org. And utilizing this platform, we have the opportunity to apply for small business research grants. And so over the past year, jointly with KIF1A, we have applied for a number of proposals. The biggest comment that we've gotten, and we haven't gotten funded yet, but the biggest criticism that we've gotten is that CAN is a rare disease, and we're a company. So commercialization of a compound for CAN doesn't seem to be profitable for us as a company. So why would we fund you? And so that has been something that was very discouraging to us. We've talked to the program officers at NIH on many occasions, and they reassured that they have a commitment to rare disease, but it has been a challenge. And so, you know, just like Wendy urged a number of you or all of you to reach out to your state senators or representatives about, you know, asking for medical coverage for genetic testing. I would also reach out to them about focusing on rare disease more. But as a strategy that KIF1A and Newsight is going to employ in the next round of research is that the KIF1A Research Network has identified a number of pathways in which KIF1A is involved in. Okay, so it turns out that as KIF1A is an essential motor protein that may be implicated in all these neurodegenerative diseases, Parkinson's, Huntington's, Alzheimer's, and ALS. And so our strategy going forward is to really focus on KIF1A still, but indicate that a successful drug or successful treatment for CAND can be extrapolated to other neurodegenerative diseases. And as Wendy alluded to, CAND is now recognized as a neurodegenerative disease. Okay, and I think the last item that I just kind of as an aside that I wanted to talk about is CAND or mutations in KIF1A has predominantly been studied in neurons, and with some transcriptomics analysis, it has turned out that KIF1A is also expressed in the support cells of the brain, the astrocytes and the oligodendrocytes. And so this, you know, this is one of the other players, you know, in the orchestra in the brain. And so Newsight is focused on generating these other cell types and then putting them together to see how, you know, potentially these mutations in these cell types might impact function and might drive a better drug discovery. And so I think I would just like to acknowledge KIF1A.org for providing the funding for all this beautiful work. The KIF1A Research Network, which provided the biology, which allowed us to develop these assays, and then obviously the CAN patients and family members, you know, the 253K and the P305L were donated by CAN patients. And then, yes. And so I think you know, Wendy pointed out the blood draws, which she's using for the genetic testing, but that's the blood also provides the cells for reprogramming to stem cells to model this disease. And I just want to highlight a couple of scientists here. Angela Murchison, she's the one that did all this beautiful work here. She generated all the neurons, she developed all the assays, and she's been essential for the progress at Newsight for the CAND program. And then secondly, Martin Nicholson, who we recruited at Newsight for a couple reasons. For the astrocyte work with KIF1A, and secondarily, his postdoc work in Taiwan was on building patient heterogeneity assays. And as Wendy alluded to, there are well over, there's so many mutations in KIF1A, and how do we capture all the phenotypes of all the patients? And we think that we have a solution, a microfluidics solution, in which we can model the patient heterogeneity in CAND. Again, this platform is in collaboration with another biotech company called Acoustic Bio. We recently got some funding for using this platform for Alzheimer's disease, but we are really exploring opportunities to use this platform for Canned. That's it. And I apologize. I just recently got cataract surgery, so I've been wearing these sunglasses, if you've noticed.
Thank you, Wayne. So I think we will try to come back to Wayne at the panel and start to move forward with our next talk. So we'll come back to this in a little bit. Thank you, Wayne.